So this is going to be a fairly simple, straightforward, hopefully straightforward explanation just to put the landscape, and like I said, we'll take questions afterwards. So I think, one of the important and challenging things is that, you're seeing parts and pieces of all this, and you're participating in parts and pieces of all this, and part of the rationale for the talks this morning is to give you a broader sense, but thinking about how it all comes together, is I guess non-trivial, and even for us who are doing the work, it's hard to think about how you align all this, and I think it must be even harder when you're on the patient side of things. So the first thing that I wanted to do is thank everybody who's participated, either through the clinic this time or other times, or through signing consent so we can look at your medical records, and the many people who are not in this room today but who have helped us, is that every single little piece of data is just such a huge help to be able to generate a broader understanding. And it's like putting a puzzle together. And if you have just one small piece of the puzzle, you can't understand everything. But once you put the whole thing together, it makes a much clearer image. And so, the purpose of the natural history study is first and foremost is to better understand the disease. We want to make sure that, if somebody is having a symptom, that we understand whether or not it's part of TUB4A associated dystrophy or not. If we can predict what kinds of symptoms are going to happen, and better anticipate them to protect the health of your children. It's also important, right, to understand how sort of big a population is and how many parts there are in that population. I think it's become quite clear to us that, when this disease was first identified, it was called HABC. We found one single genetic change. Over time, this disease has become much broader. We're now calling it TUB4A associated leukodystrophy because there's so many different types of presentations. To be truthful, we probably should call it TUB4A associated disorder because there are plenty of patients out there who don't even have a leukodystrophy because of what cells are probably affected in their brain. And so it's really important both to understand, if a TUB4A variant is found on a test, whether or not that person has a disease, but also ultimately to understand how best to target treatments. Because if you put in a clinical trial a patient who's completely different from another patient, it becomes very hard to measure the impact of a therapy, right? And so because you're not going to have the same tools to measure if the person is doing better, because if you try to compare those people to one another, they're going to look really different because they were different to begin with. And so all these concepts are incredibly important and are used by people who do clinical trial development to actually plan out what to do. And also the last thing that's important is that, there's some therapies that might need to be used that require a lot of intervention. Therapies for neurologic disorders sometimes include things like brain surgery or sometimes include things like lumbar punctures. And the last thing we want to have to do is to have to treat a patient with a placebo when you're doing something as invasive as that, just to be able to show that something works or not, right? So we just have to be really careful and thoughtful as we learn to understand this disease, that we really fully understand how people can do over time. There are lots of limitations to natural history studies. One important limitation is as you choose to participate in one, it's important to recognize they may happen years before a clinical trial is actually available. And that can be incredibly frustrating because you're contributing to the research and you have no knowledge of how much by contributing you're going to be benefiting yourself or for your child or benefiting for a next generation of affected individuals. And I think that's a hard thing to talk about, but it's a reality and one that we need to know about. I think also one thing that worries me is that, the groups may not be big enough to detect every single problem. And so even though we think we have a wrap on the disease, we might not, right? And there might still be symptoms that we don't know about because they just happen in such a small fraction of individuals that we're not picking them up yet, or because we're not smart enough to notice them yet. That's happened to me too before, and sometimes I really kick myself in other diseases saying, why did I not see this sooner, right? I think also another challenge that may happen, we were talking about the years to a clinical trial, is sometimes clinical trials are ready before we really fully understand the disease. And then we have to sometimes accept challenges and part-way answers as we're moving into clinical trials, because the last thing we really want to do is say, no, we have a good idea, we have a therapy, but we're going to wait for X number of more years till we finished our clinical trial. And that's where some of these, our natural history study, and that's where some of these approaches, like you heard Brittany talk about, where we're trying to go back in time by using your medical records, become so important. So if there's one thing that I can ask is that those of you who are in touch with other families who are not already enrolled in our medical record review, it's a pretty light level of investment for a family to participate in research. It's just allowing us to collect their medical records. It is incredibly valuable because it allows us to collect data for that we might not otherwise really be ready enough. And then the other thing too is we may find out later that we need to measure something different, and we may need to redo. So for those of you who participate in our physical therapy and occupational therapy assessments, well, we may decide, 6 months from now that really that tool wasn't working great and that we need to redo things and try a different trial. So I think what I'm trying to say is these processes are really iterative, and it's important to understand that we're all learning as we go. And although there may seem to be a big knowledge differential between those of you who are parents and affected persons and those of us who are physicians, we're actually all learning together. And, we learn an immense amount from being together with you as a group as well. Okay. All right, I think there's also a concept and words that you might use that are called preclinical research. So those things are things that Akshata talked about earlier, right? So those are the research in cells, research in animals, research on human cells, and research on human fluids, which are called biomarker studies. And those may seem, so far away from a therapy, but they're incredibly important to really understanding, like we've already talked about this morning, which cells are affected, what kind of problems are there in the cell, how can we best try to fix them. And those things, unfortunately, can take real amounts of time, right? And, I think we sometimes are, that there's a concern in me for a disconnect between, the time that the scientists need to feel comfortable to completely understand things and the time frames that you all need for us to get to therapies quicker. And I just want you to at least know that we're aware that the clock is ticking, and it's not that we're not trying to take shortcuts where we can and acknowledge that time is pressing. But I don't know how many of you picked up on Dr. Ahmed talking about it takes 40 days for this cell to grow. Well, that's a month, right? So every experiment is several months long. It takes several months for a mouse to be born and ready enough to be tested, right? Each step in this is not days, but months. And so I think it's important to understand that some of these timelines are not long because of our not working hard or our unwillingness to try to move things forward. They're just long because that's how long some of these approaches take to actually sort out. All right. And then I think it's important to think a little bit about what kinds of therapies might be applicable to this kind of disorder. And I think in particular, I think that's important because thankfully rare disease is such a focus now and is becoming such, more in the news and more present that we're seeing in the news and in the New York Times and in everything else, new therapies being developed every day, right? And certainly I feel like from me as a neurologist, it's so wonderful to be part of this field at this time, because I think where before we would have been very, open-ended about therapies, I think there are concrete examples of therapies being used and developed that make us much more hopeful, right? But I still think it's important to have at least a brief discussion about the fact that some of the therapies that you might see used for other disorders just don't make sense in this particular disorder. So first of all, there are some therapies where you can use just a medication, a pill, because there are already medications developed that work to change that body process, and we can borrow them from other therapies. So for example, some leukodystrophies are problems with inflammation. Well, there's inflammation in lots of disorders like rheumatoid arthritis and all kinds of other disorders, psoriasis, and we can just literally borrow medication that's developed for another disease where somebody else has done all the hard work already and apply it. Well, that's obviously, a shortcut, right? But not one that we can conceptually easily use for TUB4A, just because it's not something that is the same as other disorders. And the only problems that even are remotely similar are medications that are used for cancer that are high toxicity and that you wouldn't be able to use for the really long term in children like you would need to use it. So we don't have any on the shelf sitting in the pharmacy drug that at least as far as I understand right now can be used. We are trying through some of the cell models to develop things that can be measured such that drugs can be tested in case there's a mechanism we don't know about that could help. But right now, at least for a curative therapy, there isn't an easy answer. You also sometimes hear about therapies where people are being given enzymes, right? And so people think, and especially since some of those disorders are leukodystrophies, and you're going to hear more and more about this, people are being given, an enzyme to replace an enzyme that's missing. Well, that's not the problem here. So we can't just use an enzyme and give it, somehow get it in the brain, somehow make the cells make it. That's not really what the problem is, and so those are not sort of, again, on-the-shelf options. There are also a number of leukodystrophies that are treated with transplantations, right, of blood cells, and those seem to be effective, again, for certain leukodystrophies that have a lot of inflammation, because then if you change the blood cells, you change the brain's response to inflammation, and you can maybe treat things. And so, for example, a disorder called adrenoleukodystrophy is being treated by bone marrow transplant with a fair amount of success, and seems to be helping quite a bit, but that's, again, not something that we can easily just move over into HABC. You will also hear about gene therapy trials where they're putting the gene therapy into blood cells and then using that to deliver an enzyme to the brain. And that works when that's an enzyme problem where the cells that make blood can get into the brain and can replace the ability of the cells in the brain to make enzyme. But this is, again, not an enzyme problem, so not something that's not an approach that can be just borrowed and repurposed for TUP4A. One potential long-term option may be implanting oligodendrocytes directly into the brain and having them do the job that the sick oligodendrocytes in people with TUP4A-associated leukodystrophy are not doing. First of all, those are still highly experimental therapies, but they are ones we're considering. But I just want you to remember those earlier talks and at least know that that's not going to fix the neuron problem, because nobody is transplanting neurons yet, right? And so it may be that we can help only part of the population or only part of the symptoms in the population by using those kind of approaches. But those are all things that we're considering. You also, in some disorders, can use gene therapies. Well, it turns out that they use viruses to get the corrected genes into cells. Well, in the brain, the neurons do a much better job at receiving those viruses because they're much more susceptible to be infected by those viruses. And so you can typically think about those kind of problems in disorders that are primarily affecting neurons and not oligodendrocytes. And so again, we're in a situation where we might be treating only part of the disease or only part of the population. And so, those are non-trivial things to think about as you're thinking about treating enough patients to prove that something works in a clinical trial and really understanding what it does in the disease, right? And then there are also other therapies where somebody was asking, well, how do you try to turn down, that gene? If we think that the problem, which is really what we think, is that by having a mutation in TUP4A, you're basically making abnormal TUP4A that is just basically glomming up the system, right? And that if you could only shut down that gene in some fashion, that you would be able to make cells better. That's a great idea, but conceptually, it might only help certain cells. So that's where it becomes so important to have the type of models that we were discussing earlier. And just to give credit to other people who are in the room back there and who are not speaking today, because we didn't want to overwhelm you with sort of scientific mumbo jumbo. There are multiple scientists sitting in the back of the row who are each in their labs individually working with their own models, with different animal models and with different cell models to try to answer some of these questions. And so there's multiple simultaneous threads of exploration that are all happening, together, and we all decided together as a group that it was kind of nice that we were doing almost the same thing in multiple places because it provides greater depth and greater experience. But it's going to take a while to sort these things out because of those complexities with multiple cell systems, right? And again, I know that we're very aware that this is a clock and that we are all, trying to make things move as quickly as possible. And I just wanted to reassure you that we're not sensitive to that, but we also need to make sure that we answer the questions we need to safely and effectively do this. All right? So I think if you could all write your questions down, we will discuss them at the round table, right? And then right now, we're going to move forward to the next series of talks that are more clinical, right?